Well, then welcome to the third unit of this module 3. It is aimed at the calculation and evaluation of key breeding parameters. I will start with a more theoretical conceptual approach, and my colleague Barbara from CAIS of Slovenia will provide a more applied or practical approach. Okay. Anyway, we decided not to go in detail with these parameters and breeding parameters because it can be very difficult to understand. Okay, I've been a student years ago, and I remember that if you go further and in detail with the breeding parameters, particularly in quantitative traits, it can be a total nightmare. Okay, so in order to give you provide you the main issues and in a very easy way, we have simplified a lot the main breeding parameters, in particular those related to quantitative traits. Okay, so if you want to go in detail or to learn a bit more related to these issues of key breeding parameters, I encourage you to go to visit or download the available materials, additional materials, or the books aimed to plant breeding recommended in the units 1 and 2, because they are quite comprehensive, not only for generation of new diversity, but also these books are highly recommended for breeding strategies in plants, in different reproductive systems, etc., and even for the estimation of the key breeding parameters. So if you want to go in detail to learn more about key breeding parameters, follow the additional training materials at the end of this unit, but also you can go to the books recommended for units 1 and units 2, okay, of this module. Well, I will start now with the unit 3, calculation and evaluation of key breeding parameters, presented by myself and my colleague Barbara Pipan. Always repeating, I'm very fast. We are on the context of the organic plant breeding training initiative of the LiveSeeding project. Okay, I remember to all of you that there are also other training opportunities, courses, and materials in the training part of the LiveSeeding webpage, and I strongly recommend you to have a look and to find and ensure something that will complement this organic plant breeding training and will be of interest for you. Okay. Well, as I told you at the beginning this morning, we are in the module 3. Still module 4 and module 5 are coming, which will be launched at the beginning of March 4th and March 7th days, okay, in less than 1 month. But today we are in the module 3, breeding methods fundamentals. We are now, okay, sorry, this slide corresponds to the unit 2. Actually, we are on unit 3, 3.3, calculation and evaluation of key breeding parameters. It will take from now till about 4 p.m., 5 minutes less, 5 minutes late, more or less. We will make up half an hour break from 4 p.m. to 4:30. Okay, and then we will finish with unit 4, aimed at fundamentals in participatory plant breeding, with my colleagues Pedro Méndez and Veronique Chabla from Instituto Politécnico de Coimbra and INRAE, respectively. Okay, coming back to the current unit, unit 3, calculation and evaluation of key breeding parameters. Okay, as always, a fast summary or index of the things you are going to see. We will provide a presentation about the main topics or kind of traits, ways and parameters to analyze or to estimate, about 60 minutes. I will start with a comparative between qualitative and quantitative traits, a short view about the analysis of qualitative traits and quantitative traits, okay? And then I will give the floor to Barbara, who will provide info, more practical info about parameters and traits of interest during breeding workflow, a more applied approach. Okay, I will provide more or less the more theoretical basis, and then Barbara will provide a more applied or practical point of view. Then we will finish by a fast quiz as in all the units, and we will finish in, depending on the time left, a short debate, grow up, revision of last questions, and the proposed homework for this unit. Okay. As always, two important things: the fast quiz and the proposed homework must be done if you want to get certificates of attendance. And also, please send to Barbara, and particularly to me, and very important, don't forget Petra's mail, who is centralizing all the quiz and the homeworks for checking. Okay, for the quiz is now during this unit. For the homework, you will be leave 1 week. So you must send the homework by next Thursday, 20th. Okay. And the questions is important, as in all the units, please through the chat, because this is more efficient to manage it this way. Petra, in case we forgot or we missed some of the comments or the questions, Petra will manage and will remind us the questions still to be answered. And if we have no time during this session, during this lecture, don't worry, because we will revise and provide the answer in the next days. So let's start by the beginning, in a redundant way, qualitative versus quantitative. Okay, many of you should be familiar. You have some basis of plant breeding or genetics, but even for you, and particularly for those who are not so familiar, some concepts are in some corner of the minds. Okay, I will revise very briefly and in an ordered and summarized fashion what are qualitative and quantitative traits. Okay, qualitative traits are, you can recognize this face, considered the Mendelian traits. Okay, they are also called the Mendelian traits because Mendel, during his studies, was very lucky because he was working with qualitative and very simple traits and highly inherited traits. Without these traits in peace, probably Mr. Mendel had not discovered the mathematical rules to manage the basic inheritance of simple traits. Okay, these qualitative traits are those traits where the individuals can be clearly classified and distributed in categories. Remember the Mendel's piece. The seeds, the piece could be green or could be yellow, could be hard epidermis or could be soft epidermis. The color of the legume, green or yellow. The color of the flower, white or purple, etc., etc. Okay. Within this context, many important traits in breeding, particularly resistances to diseases, are usually qualitative traits. Okay. These qualitative traits are considered monogenic or oligogenic traits. That means that their inheritance is controlled by just one gene or very few genes, or a mixture. One main gene, which explains most of the differences between the individuals in inheritance terms, complemented by the small effect of minor traits. But on the whole, most of the variance, most of the differences among individuals in segregating populations can be explained by one main gene. This is the case of the traits, big traits Mendel's worked with, and many resistances to diseases in plants. Okay. On the contrary, we have the quantitative traits. For you to identify clearly which ones are these. They are the traits which are measured. Okay, but in contrast to the qualitative traits, which where individuals can be clearly classified in categories, you are red, you are yellow, you are pale yellow, or you are red, you are pink, you are black, you are white, and there is not other possibilities, clear categories. Quantitative traits have no categories. I mean, quantitative traits are those traits that are measured. Okay, measured, based on metrics. In fact, historically, this part of genetics, the inheritance of quantitative traits, has been called biometrics. Okay, in these traits, the individuals of a segregating population are not distributed in clear categories. On the contrary, they appear distributed continuously. For instance, quantitative traits like yield. Yield is measured in terms of kilograms or grams per plant, or in an agronomic approach, kilograms per square meter or tons per hectare. Okay? Can you establish natural categories like the qualitative ones? No, you cannot. Of course, you can create artificial intervals of the yield. Okay, individuals from 0 to 1 kilometer to 1 kilo per plant, individuals from 1 kilo to 2 kilos, from 2 to 3 kilos, etc., etc. But this is not the real life, because some individuals have 1.65, others have 1.88, others have 2.15, etc. So the most realistic way to represent this kind of traits is continuously using continuous functions. The most common ones are the normal distributions, as you can see here. Also, in contrast to qualitative traits, quantitative traits are considered that their expression depends on several or many genes. So they are so-called polygenic traits. After the rediscovery of the Mendel's laws and the mathematical rules to explain inheritance of simple or monogenic traits, there was hard discussions about if quantitative traits were inherited or not according to the Mendel's rules. It took several years, but at the end, geneticists for qualitative or for quantitative traits agreed that also quantitative traits are inherited according to the Mendel rules. But the difference on quantitative traits is that they are controlled by so many genes that on segregating population, the categories can be decens or cents. Okay, so they are distributed in so many categories that it is almost continuous. In addition, this quantitative or polygenics or measured traits suffer more than qualitative traits the effects of the environment. So even individuals that genetically could be in category, let's say 1.25 kilos per plant, the environment can put in the distribution on the 1.35 or 1.40, while other individuals which genetically could produce till 1.95, the environment may decrease their final phenotypic expression to 1.45, for instance. So, in addition to this complex distribution of so many categories because of so many genes controlling the expression of segregating populations, the environmental effect modulating the final phenotypic expression also works on that. Okay, so this makes the difference between the distribution of monogenic and polygenic traits, qualitative and quantitative traits. And now we are going in more details. Qualitative traits, to be analyzed, is or geneticists and breeders work in a very simple way. Okay, you must remember that the individuals of segregating population, the ones who are key to define the inheritance of a trait, we can say the color of the piece of Mendel, but we can also say how is controlling this disease, the resistance to this disease. They work with the number of individuals on each category. Okay, think that we have three categories for the expression of this resistance: tolerant, mid-tolerant, or susceptible. And there's no other expression. Or you are yellow peppers, orange pepper, or red peppers, and no other categories. So you are working with the number of individuals within each category of the trait. Then, to analyze and to work with these traits, geneticists and breeders check the inheritance model statistically with the frequencies corresponding to each category. And to work with this kind of data, number of frequency of individuals per each category, there are two main statistical and mathematical functions to work with. Chi-square, that probably is quite familiar to many of you, is used for qualitative traits to check the different inheritance model you propose to explain the control of your trait. Okay, is this a dominant control? This is a in codominant inheritance. There is no dominant, or this trait is not actually a qualitative trait, but it's a quantitative trait that cannot be explained with the Mendelian models or the complete dominance or the codominance models. Okay, this response is given through chi-square methods. And also, to work with these quantitative traits, geneticists and breeders work with the binomial distributions. Okay, working with the number of individuals expected for each category and the P or Q, the frequencies of expected, the expected frequencies for each phenotype, each category within the trait. So you will see that in the case that one trait is studied, or in literature, their study, their inheritance analysis is performed through chi-square and binomial functions, without any doubt, you are behind a qualitative trait. Okay, I did not enter in how it is estimated, the chi-square and binomial functions, because the formula is quite common, and the binomial, you have here the formula, and Barbara will provide practical examples later. Okay, I will put, on the contrary, more emphasis in the quantitative traits. Okay, then clear the qualitative traits, simple traits controlled by one gene or very few genes, very few effect of environment, and if any, it is quite easy to detect. Now we go to quantitative traits. We are back to quantitative traits. How are they studied? Remember that these traits are measured, so no natural categories are possible. And I repeat, also, as many genes can be involved in their inheritance, the environment may modulate the final phenotypic expression, creating, let's say, mirages and hiding the real gene functions. How these traits are studied, or which parameters are used to represent first the population. Remember that these traits are measured and they are distributed in a continuous way. The most usual way are the normal distributions. And for this, the most basic and easy to represent what happens with a population, a segregating population in terms of inheritance, are the mean and the variance. With the mean, have a look to the picture in the middle. With the mean, we know the general value of our population. Okay, this population is 5.5 on average. Good. This is a good parameter to define, but we need another one, which is given by variance, because if you see the picture in the middle, you have two distributions for two different traits, where the average value is 100 for both. Okay, but you can see that the population are not the same. The red population will represent red individuals for the red trait, are concentrated in a very narrow variation interval around the mean of 100. On the contrary, the other population has also 100 on average for the value of the trait we're interested, but you can see that the individuals are distributed widely, in a wider way. So we have two populations speaking of the same trait, like yield, for instance, 100 or fruit size, 100 grains per fruit, but on population one, the variation is very narrow, while on population two, the blue one, the variance, the diversity is wider. So now you can understand that to represent in the most accurate, but also in the simplest way, a population, we need the mean to have a look, to know on the general terms the value, the general value of the population, but also the variance, which give us the idea of how wide, how diverse is the population around this mean value. Okay? Using these two main and easy parameters, breeders and geneticists estimate a range of biometrics and quantitative parameters. Today, I will just mention a few: the coefficients of variation, covariances, correlations, heritability, and the study of the gene genotypic by environmental interaction. But there are many more. But as I told you, here in this presentation, we will be as simple as possible for you to put in the context and understand properly the main aspects on quantitative traits analysis. Okay, coefficients of variation. It is also a simple way to estimate the diversity of a population. Instead of managing big numbers with the variance, geneticists and breeders of quantitative traits use the coefficient of variation as a percentage, and is estimated by dividing the standard deviation, which is the square root of the variance, divided by the mean. And then this ratio is multiplied by 100. Then you have a coefficient of variation in percentage, and you represent in a very simple way how diverse is your population for this trait, based on the ratio between the standard deviation and the mean. Also, the covariance formula, which are on the bottom of the left corner, it's used to estimate several parameters, several inheritance parameters, but particularly a very important parameter useful for breeders and geneticists of quantitative traits, which is the Pearson correlation coefficient. The correlation coefficient establishes, here you have the formula. Okay, I must revise because I did not copy properly the formula. I will provide you the revised version, sorry. This, but anyway, the correlation express the way two traits in terms of trend, positive or negative trend, and magnitude, the way two traits under the supervision of a breeder change with the other. I mean, if two traits, trait X, trait Y, are positively and strongly correlated, you can see the examples on the right. First example on the upper part, a strong positive correlation. That means that high levels, high values of the trait X are highly related with high values of the trait Y. Okay, and also the contrary. Low values of the trait X are highly correlated with low values of the trait Y. Maybe positive correlation also exists in your, also appear in your experiments between these traits X and Y, but sometimes they are not so strong. It is positive, but not strong. Weak positive correlation. You can see that still the correlation exists, but for instance, high values of the X trait correlate, yes, in a positive way, but not at high magnitude with Y trait. Okay. And on the contrary, you have the examples of negative correlation and strongly negatively correlated. Is the example of the right, that the lower, the higher the value of the X trait, the lower the value of the Y trait. Okay, you have also with negative correlations, and also you can find examples of no correlation. Some occasions, the trait X and the trait Y are not related. The values for each individual on the values of X and the values of Y are not clearly correlated. They are not dependent one each other. Okay, so you can see that traits can be correlated in positive way or negative way in the magnitude hard or weakly, or even you can find traits not correlated. Why this parameter is important for geneticists and breeders? Okay, think you are a breeder that is working on yield. You are interested in increasing yield as much as possible, but also quality or sugars or taste of your fruits are important. And imagine that both traits are negatively correlated. This is important to know, because if you are too strict selecting those individuals with the highest yield, probably you are losing quality in your fruits. Perhaps you don't need to be so strict selecting the most productive individuals, because you are also interested in as satisfactory, not excellent yield, but ensuring a minimum level of sugars or good taste in your fruits. Okay? So this is important to know, and this is an example of why correlations are important for breeders. How traits are related within your breeding populations. Well, and now probably the most important trait in quantitative traits, which is heritability. It, as the term says, heritability, on the whole, this parameter is used to explain to which extent the phenotypic differences you see and record in your population, which will be submitted to selection, these phenotypic differences are based or have genetic basis, are based in genetic differences. So you can see and record that in these 1,000 individuals which will be submitted to selection, I can see a mean of 3 kilos per plant on average, and a variance of 6.5. Okay, but this variance or these coefficients of variation, these phenotypic variant differences and variances, to which extent depend on real genetic differences? Because this is the key. You will select based on what you see and with your record, the phenotypic differences. But if the phenotypic differences have not a strong genetic basis, the response to your selection will be very low. Okay. On the contrary, if phenotypic differences have a high support of genetic differences underlying, I mean, the individuals which look good at phenotypic level is because they have good genes in comparison to the other individual with look not so good phenotypically. Okay, this is important because when you perform selection, the only part will be inherited from one population to the next based on the selection. Only the genetic differences are inherited. So if you want to have a high response to selection, it is essential to work with high heritability traits. Okay, you can see in a schematic way how the expression of a trait the phenotypic expression depends on the genotype, the environment, and depends on if your traits are performing in different environments, the genotype by environment interaction, which in terms of differences, in terms of variances, goes to the formula in the bottom, which is phenotypic variance equal to genotypic variance plus environmental variance plus, if applicable, genotype by environmental variance. Okay, and on the whole, the ratio between the genotypic variance against the rest of the phenotypic variance, which is the whole variance, is the key and is the basis for heritability. The higher the value of the heritability, the higher the genetic basis of the differences you are seeing in your population. The phenotypic differences you are seeing, and probably the higher the response to selection. Okay, so in a bit more detail, we can distinguish between two kinds of heritability. Broad sense heritability, which comes by the directly dividing, or the ratio of genotypic variance divided by phenotypic or total variance. In such case, this heritability is useful when all the genotype of the selected individuals or the genotypic part pass from one generation to the next. I will explain later with a bit more detail what does this mean. And we have also narrow sense heritability. In this case, narrow sense heritability, the part of the variance which is in the upper part is the additive variance divided by the total or phenotypic variance. It's a more strict, it's a narrow sense heritability. Okay, and usually it's smaller than the broad sense heritability. In this case, the case that where narrow sense heritability is needed, in this case, only the additive part is considered. The additive part, which explains the differences among the individuals, is considered and passed from one generation to the next. And this is the reason for which is the only part considered in the ratio against the phenotypic variance. The dominant part and other genetic interactions are not considered because they do not pass from one generation to the other. Okay, here you have short explanation for that. Broad sense heritability are mainly used when you are working in terms of clone selection or performing selection among highly inbred or autogamous lines. When you are evaluating several clones and you will perform a multiplication of those clones which will be selected for the next year, imagine you are evaluating 10 clones. Reply it 10 times. So 10 replicates, clonal replicates of each clonal variety. And then on these 10 clones, you see that clone 3 and clone 5 are the best ones, okay? For the next step, or the breeding program, you will take again replicates of this clone 3 and clone 5 in the next evaluation. What are you doing? You are replicating exactly all the whole genotype of this clone 3 and clone 5. Therefore, all the genetic effects, the additive, the dominant, the genetic interactions among genes pass completely from one generation to the next. Okay, because you are replying in a clonal way. So the whole genotype pass from one generation to the next. Or even when you're working with inbred lines, usually in highly autogamous species, they are sexually reproduced, yes, But for the traits of interest, the genes are already fixed, homozygous. So if you reply sexually by selfing, which is the reproductive system of this species, one inbred line, the offspring is exactly the same because the genes are homozygous, and selfing-homozygous genes keep the same in the next generation. So broad sense heritability is useful for clonal selection or working with sexually but highly autogamous or inbred lines. Well, by contrast, narrow sense heritability, which only considers the additive part, is mainly used for sexually reproduced species, yes, but whose populations are allogamous and or open-pollinated. I mean, populations who still are segregating, highly heterozygous, a mixture of individuals, which has a lot of genes in heterozygous fashion. Other genes can be in homozygous fashion, but there is a mixture. Okay? Imagine an allogamous population, segregated. You have the parent population, which are an average of x sub 0, okay? And the distribution, the normal distribution of the different individuals, okay? Imagine that you select only a part, the best ones, based on your phenotypic data. You are selecting just the part on the right, which represent 5-10% of the original population, the 5-10% of the best individuals. And this subpopulation, this good population, has a mean of XS, corresponding to the selected population. Okay? Well, imagine that there are 1,000 individuals in the original population, and you select 10%, which means 100 individuals. These individuals are highly heterozygous. Other genes are homozygous, but the mixture within each individual is quite high. Okay, these individuals are selected, then they reproduce, open pollinated. So imagine individuals that are quite heterozygous, exposed to open pollination, how their offspring will be. There will appear to the difference of clone selection or highly inbred or autogamous lines, the difference is that in this case, this selected population will suffer from one generation to the other to a high level of recombination of the alleles. So the only part which will pass from one population to the other, from the selected population to the next generation, to the offspring of this selected population, is the additive part. I mean, the good alleles, the good genes that these selected individuals had at the beginning. Okay? Imagine that the alleles number 1, A1, B1, C1, D1 are good for the expression of the trait. Okay. The only thing you are sure is that from these 100 individuals you have preselected, which are submitted to new recombinations, is that, okay, these 100 individuals had a bag or bring a bag of 500 genes favorable with the number 1. But I don't know how these genes will be reordered, will be recombined in the next generation. Okay, this is the dominant part, which is not inherited. This is the reason for which when you evaluate the offspring of this selected population, the mean of the selected population is not as good as that of the parents. They suffer some regression. Why? Because the specific gene combination of the parents have not been inherited. They have suffered a recombination, and only the additive part has passed from one generation to the other. In addition, when you select some individuals, some of them may the environment may have caused a marriage with some of them, and therefore they were not really so good. So you are introducing bad genes on the pot, and at the end you have a new offspring, the offspring generation, which is good, which is better than the original value of the original population, x0. Now we have x1, but not as good as the parents were. Okay, this is a clear example on how only the additive part and the pass from one generation to other, and also take care about the environment when you perform the selection. In these cases, the irritability can be estimated in the following way. First, you estimate a simple parameter, which is selection differential, which is the difference between the mean of the selected parents against the whole mean of the population under selection, under evaluation. Okay, this is selection differential. And then in the offspring, you estimate another parameter, the response to selection, which is, again, in an analogous way, the difference between the mean of the offspring from the selected parents against, again, the mean of the original population. This is the response to selection. And then the irritability in narrow sense can be estimated as the ratio, as simple as the ratio of the response to selection against the selection differential. It's very simple. I put a selection effort in the previous generation, which is the selection differential, and what have I gained with the selection? With what has been the response to selection in my offspring, the response. And this ratio is a very simple way to understand in which extent the trait is inherited. Okay. And last but not least, it is important to know that inheritance move between 0 to 1. The closer to 1 the value of the heritability of your trait, the higher the inheritance. And the most controlled by genetic differences is your trait, and better for the breeder, because you will have a high response to selection. On the contrary, the lower the value of the heritability, the lower the contribution of genetic differences and higher contribution of the environment. And therefore, in such cases, maybe you are thinking that you are selecting good individuals, but on the contrary, the environment is creating a mirage. Okay, and you are selecting mirages, okay? Because the phenotypic variation is mostly dominated by the environment, by environment differences, and very few by genotypic differences. Okay, so between 0 and 1. 0, bad for the breeder. Closer to 1, good for the breeder, and to have a good response to selection. Okay, and finally, the genotype by environment interaction. I have simplified very well to understand this concept. Imagine you are working with a battery of varieties working in several environments. In this case, I have simplified 2 environments, environment 1, environment 2, and variety A in blue and variety B in orange. The lines represent, in the case of variety A in blue, the value of this variety A in the environmental one, which is 2 kilos per plant, for instance, and in environment 2, which increases to 6 kilos per plant. Okay, you can see the increase from environment 1 to environment 2 of variety A. Change from environment 1 to environment 2 increase the behavior of the variety A. And the same occurs with variety B, which achieved 4 kilos per plant in environment 1 and achieves up to 8 kilos per plant in environment 2. Okay? In this case, we could say that there is environmental effect. Changing from environmental 1 to environment 2 increases yield clearly in the two varieties, but in the two varieties in the same level. In such case, there is no genotype by environment interaction. Okay? And on the examples on the other side, let's see to the far right example, you can see that the performance of the two varieties are crossed. Variety A increase from environment 1 to environment 2, clearly. By contrast, variety B decreases when you pass from environmental 1 to environment 2. What is happening here? Can I say that one environment is better compared to the other? No, because one increases the yield in one variety and the other decreases, and the contrary, one environment to the other. Okay. In this case, we have genotype by environmental interaction. Okay. When some varieties behave good in one environment and bad in the other, and the ones who performed well in the first environment were bad in the first environment and good in the second, this is a crossed effect, changing the environments, and then there is genotype by environment interaction. Okay. And what about the last example in the middle? Is there genotype by environment interaction? I can say yes. Of course, it depends on the statistical terms, but yes, because we can say yes, but the change of environment from 1 to 2 improves the level of expression in both varieties. Yes, but not at the same magnitude. The lines cross, and you can see that variety B, yes, is increasing, changing from environmental 1 to environment 2, but the increase in variety A is still stronger. So this is a case of genotype by environment interaction in magnitude, not in trend, okay, but in magnitude. This is very useful for breeders because when they work with a huge collection of varieties, it is important to know first how each environment increase or decrease the expression of the trait. Maybe it is clear this environment is better for the expression of the trait in all the varieties. Okay, this is clear. But maybe the change of environment is not so good for all the varieties. It's good for one, for some, and bad for others, and the contrary when you change the environment. In this case, breeders must work with exploding the genotype by environment interaction. That means that they can take advantage of this phenomenon, selecting specifically the varieties which best behave on each environment. This is the exploitation of the genotypic by environment interaction. Okay, and this is very important for breeders. And I have finished my part, and now it's time for Barbara. Barbara, I have finished the more theoretical part. Now it's time for your part, the more practical appliers. So please, the floor is yours. Just let me know when I have to pass these slides.
Okay. Thank you, Adrian. Good afternoon to everybody. So, and thank you, Adrian, for a really nice theoretical background of all the parameters and key breeding parameters which are highly appreciated in the breeding program. So now we will move to the common bean example. So before lunch, I present the common bean breeding program held at Agricultural Institute of Slovenia. And here in this presentation, we will go through our actual results actually and data and traits of interest which are recorded through our breeding program and from which then the key breeding parameters can be calculated from. Please, Adrian, could you move the... Okay, so as it was already said by Adrian, that key breeding parameters are essential metrics and concept used in plant breeding to evaluate and improve the traits of interest. Of course, before we were talking about breeding objectives and traits of interest. And here actually we have different breeding objectives, and that's why we have as well created different groups of breeding objectives, and within the same group we have the same breeding materials to be selected following those objectives, so that we pre-prepared actually before pre-prepared the groups of breeding objectives when the selection is done through the breeding. And before we can calculate and evaluate key breeding parameters, we have to collect and record the results, the data about different traits on different levels. So here we have on phenotypic traits, or we go on the morphologic level, genomic data, genetic data, if they're available, then biochemical data, nutritional data, different results of resistant tests and observations, and of course then the seed quality parameters, and put all those traits together for the same group of breeding line from, I don't know, the same combination of the similar combination with the same breeding objectives. And when we have all those data collected from different levels to put together, it's then up to us to decide which breeding line from, I don't know, 20 most promising ones at the end is this breeding lines which is the best performing on all different levels. And this is quite complex thing. If you can imagine that we record the data and the results on the field in the screen houses, we have the laboratory results from marker-assisted selection, and then we have to combine all the data together to put them on the common denominator and then finally select the best one. So with this, I would really like to go through this workflow very briefly, but just to provide you an insight how the data and the results at the end are organized and how do we do at our institute these calculations at the end, because the common goal for the breeder at the end is to select the elite breeding line, which is then put to the registration. So you can imagine, so I mentioned how many traits of interest are recorded on how many levels, on how many places. So, and then different environments, and as Adrian exposed, which is very important because maybe earlier I didn't mention, we have different locations where we evaluate our breeding lines, not just this our location, which is where we have breeding materials, but also the other in Slovenia, that we can compare different environments and how the most elite breeding lines are behavior in different environments. And this is very important. So that's why it is important that we, at the end, that our decision at the end is supported with the data, with the statistics, and that everything is actually put and select on the correct way. So please, Adrian, could you move the slide? Okay, so in module 2, Adrian was talking about descriptors. So descriptors are very important for the evaluation of the germplasm. So it's the evaluation of the germplasm of different plant species, so as well as for the common bean, and it is also very important for the evaluation of the breeding materials as well. So here I put different descriptors, which are actually descriptors and the traits already kind of formally established for the common bean. Some of them are really old, so older than me, and some of them are actually recently published, and they're a kind of a group of traits of interest which are followed through the our breeding process as well. So having this in mind, and having in mind that at the end, when we will put our selected elite breeding line into the registration, we will have to provide data about this elite breeding line, and this breeding line should be very well characterized and evaluated as well, so that we submit the technical questionnaire with all the details about the traits of this breeding line. So with this, I can say sometimes that for our breeding lines, before they're put into the registration, we know much more about those plants that we know about ourselves. So because they're really very well characterized. And now we will go through the groups of traits which are recorded during the breeding program for our breeding material. Adrian, could you please move to the next slide? Okay, so first one is I put the name morphodata, so on the morphologic level. So these are the descriptors, traits of interest, which are already established in the descriptors which were presented on the previous slide and are associated with different traits. And those descriptors are recorded during different F generations when the material is growing, is selecting, and of course the characterization and evaluation is done more in details than in F5 or F6 generation, when we reach the desired level of stability and uniformity of the population of the same breeding line, because this is, as we heard before, required for the DUS testing. And along with these traits and these descriptors are associated with different part of the plant. So it's actually there are traits which are associated with the habitus, general habitus, associated with leaves, associated with pods, associated with flower, associated with seeds, and at the end, of course, associated with yield. So these are those who are colored in orange color and associated with the, I don't know, yield per plant, and then associated with the visual score about the susceptibility to the diseases, pathogens, and which is the most important. So in our case as well, so this is a kind of our know-how in the breeding of common bean, is that breeder score. So I put it here with a tick. Breeder score is very important because the breeder go through all the materials, through the vegetation for at least 3 to 4 times, and to evaluate the general performance of the plant. So what, of course, everything according to the breeding objective. And this breeder score is something additional, which is also highly appreciated and evaluated through our breeding program. Along with those characteristics which are put here, of course, photomaterial is very desired as well, and should be also photo documentation is very important within the whole procedure and breeding progress, including the selection, because it's really nice that you have everything also photo documented stated that you can then think about some traceability and the performance in different generation of the, I don't know, the most promising breeding lines through the whole breeding procedure. Could you please move, Adrian, to the next slide? Then also today I talked a little bit during the presentation of the breeding program about the genetic background. Genetic background is very, very important. So in our case, we have a panel of 24 different DNA markers, different types of the DNA markers, which are associated with some traits of interest. So as Adrian very nicely explained before, so some of the traits, of most of the traits, especially quantitative ones, are polygenic. So a lot of different genes are included into the inheritance of such traits. And this is very important that we have, let me say, the most important markers which are associated with the genes, and then to screen them and to screen then our breeding material in the F2 generation when the segregation diversity is the highest, to help us with the selection and to a kind of do the complementary selection with the phenotypic selection. And those kinds of data, so data on genetics, are then also evaluated or processed differently, because it's we are using the tools of population genetic for the population genetic analysis and algorithms which are associated with that. So parameters of the genetic variability, informativity of the markers, and the population structure, and everything on the basis on the molecular data. So this is really a kind of a separate field of the evaluation of the genetic background, which will be explained in module 4 in March, in 4th of March, more in the details. So please, Adrian, move to the next slide. So we are continuing with the next group of the traits, which are as well very important. So it's pathogen resistance. As far as the technical questionnaire for the common bean registration of the variety is requiring the information about susceptibility to the Xanthomonas, Pseudomonas, Coletotrichum lindemuthianum, and the common necrosis mosaic virus, we have to provide the information as well on that. So this information is also important. How do we decide on the material from the most promising material, which elite line will be selected then for the registration? And this is something actually very important part of the evaluation, because our breeding objectives as well following the direction that our varieties have some tolerance or resistance to especially diseases which are most important in common bean. Next slide, please. Okay, then the next group of the traits of interest which are following and recording during our breeding program and done on our breeding materials is biochemical composition, and as well the organoleptic data and the results of this part as well. So here we are focusing about contents of microelements, macroelements, proteins, sugars. Sugars are more important in pots, and a percentage of inhibition of trypsin and trypsin inhibitors content. So there are different parameters, biochemical parameters, which are also considered for the final selection of our materials. From the group of the organoleptic traits, we are also measuring the the duration of the cooking, so that the pods and the seeds are cooked, and the stringless after cooking in the pods, and the taste, the general taste. So different traits are associated with that. So my family is very well familiar with this, because we are doing this also very frequently to test our breeding material. Okay, so now we will move to the next group. So please, for the next slide, Adrian. Okay, so this is the I would not say important, but something which is not highly required or required for the to be involved in the multi criteria decision at the end, multi criteria decision analysis at the end, but very important as well for the providing the final data for the submission of the variety to the registration for the community plant variety office technical questionnaire. So this is the germination rate, the germination vigor, absolute 1000 mass, and here in the bottom you will see with the report from our ISTA accredited seed testing laboratory. It's in Slovenian language, but nevertheless, do not bother with that. It's very important that we have also the results of this analysis as well. Okay, so please, Adrian, move to the next slide. Okay, so now we have we went through all different main groups of the traits collected through the whole breeding process. Okay, so now we have, I don't know, 25, 30 the most promising breeding lines, which are evaluated and characterized on different, very different level. So to put everything together, so now we have a lot of data, and now we have to decide which one is the best, or I don't know, which three are the best. Maybe if we would like to do some additional tests, additional open field performance to see the behavior and to do the final comparison with the standard varieties, and maybe are not distinctive too much between each other, or for some reason we have to decide which one is the best. So Adrian very well explained the quantitative traits, the qualitative traits, the background of those traits. And in our case, for this statistical evaluation or statistical analysis and data analysis, we're mostly using R programming environment, but it's not important which program is used. We are using R because all different packages, you can combine it with different packages to do the different types of analysis, and you can be more flexible, but all the other statistical programs have the same algorithms and the same formulas implemented, so it's not important. Important is the measure, what you want to calculate, and what this statistical measure stands for. So it was already mentioned that descriptive statistics, which is basic statistics, is important also to get an insight and an impression on our material. So in this table on the top right, you will see an example about the different agromorphological parameters, biochemical parameters, which were measured on flowers, on pods, on seeds, and different traits, so qualitative traits, quantitative traits, and the different units of measures. And then we provide the minimum, the maximum, the mean value, the standard deviation, the coefficient of the variation, and as well the Shannon viewer diversity index. So these are basic descriptive statistics which are allow us the insight into our breeding material, which is organized in our case always like this, that it is from the similar combination and the common breeding objective, so they can be compared. Okay, And I also mentioned the population genetics analysis, that this is specific and it has a different type of data and as well as results, and then it should be transformed correctly into the way that it can be compared in the multi-decision analysis at the end. And then Adrian already mentioned the frequency distribution, and for the estimation of the diversity levels, those measures are important to evaluate the variability of our materials. And then, of course, very important, it was also mentioned by Adrian, is Spearman coefficient, who allows us to assess the pairwise patterns of association between traits. So there can be positively correlated or negatively correlated. So it's very important. And then to calculate grower distance, to estimate the similarity between pairwise breeding materials based on the all traits analyzed. So it's very important that we have combination trait by trait, and that we have combination of all traits analyzed for our breeding material or subset of the breeding lines, which are the most promising ones. Okay, Adrian, could you please move for the next slide? Okay. And then more advanced thing is factor analysis of mixed data is applied to investigate the differentiation patterns between breeding materials and to identify the main factors responsible for this differentiation. So this is very important to see which traits influence the most to the variability and differentiation of the breeding lines, so which has the higher impact on the variability. And then, of course, employing the hierarchical clustering based on principal components, which is commonly used as a complement for factor analysis, to further categorize breeding materials into more specific groups. So if we have breeding material, which is in our analysis already organized to have the same breeding objective, to coming from the similar combinations of the traits and coming from the similar parents. So it is very important for this analysis that we see the grouping. So if there is some, I don't know, some subgroup or some different group of the breeding lines which might deviate from the breeding objectives which were already stated, and maybe for these are interesting, I don't know, for the other objectives for the breeding to improve for the other traits. So it's very important that we have I would say eyes open and to see all those deviations, and to maybe highlight some genotypes, some breeding lines which are different and are there with the specific reason, and also as well good, but maybe for the other breeding objective, not the one who we're searching for. And then at the end, of course, we are using a comprehensive multi-criteria decision analysis to evaluate and rank breeding materials based on their suitability for the final selection. So the results of this analysis is in composite scores reflecting the overall performance of each breeding material. And this is the Composite Performance Index, CPI. And this index is very important. So to get this index, it's very important before that, as I mentioned, we rank the traits of interest according to the importance of those traits. So if I say that, I don't know, the most important is yield per plant, I will rank this as 1. And the second of importance is the genetic background for tolerance to water stress will rank with 2. And then like this, and then we combine those traits, all traits which are available and data and results available within the same analysis, to which allow us to select the best breeding line. So maybe just to mention, but not bother you with this. These traits are this multi-criteria decision analysis included qualitative traits and quantitative traits as well. But quantitative traits should be normalized first and go through the special procedure, and then that they are all again on the common denominator to be compared between each other. And at the end, finally, in this table in the middle, in the red, we have the winner. So we have the most, let me say, the best performing bleeding line according to the our decision multi-criteria decision analysis, and selected actually for the registration. So if everything went smooth and all the data were available, then we decided that this breeding line is the best, and with these types of analysis and support of the statistical analysis and results, and of course considering key breeding parameters, is the best for the registration. So could you please move to the next slide, Adrian? So these are some articles maybe for the deeper reading and to understand more in details the result of the genomic analysis to result what was I was explained about. So it's actually from the results from the breeding program and breeding procedure, and how then the best performing line could be identified. So with this, I would say, maybe isn't there another slide, Adrian? I don't maybe. Okay, so we are with quiz. So yeah, with this, what are just to wrap up and maybe expose the main challenge for the future is that maybe just to point out that to calculate and evaluate key breeding parameters is highly complex, covering different types of data and results from different years, from um, different different locations within breeding procedure, and that key breeding parameters are crucial for the final selection, are crucial in terms of being effective and make a good decision in breeding programs. And at the moment, our main challenge is to make the breeding process, the whole breeding process, our breeding materials, data, and the results from the breeding process digitalized, and to make key breeding parameters be automatically calculated from the data within the breeding workflow. So with this, we have a kind of a national project dedicated to that. It is called the platform Kis Digi Fizhou. So it's in Slovenian language. Fizhou in Slovenian language means common bean. And we started with that because you can imagine that the 10 years which are needed for the from the first crosses to the final registration of the varieties, a long period, and a lot of data and a lot of results are collected and recorded, and should be organized, and it's very important that it is organized on the correct way and that is traceable. So that's why this platform, we hope that will help us to reach this goal as well. And as mentioned before, so I put also some logos of the related projects ongoing at KISS, at our department, in association with the legumes, with the common bean. So with this, I would really like to thank you for your attention and leave the floor to Adrian, since it is time for the short quiz. So thanks, and Adrian, please.
Okay. Thank you, Barbara, for such a good synthesis of practical applications of breeding and then key parameters. Quite nice, and I think it will complement the theoretical basis with such good numbers. And now it's time for the quiz. I can leave you. I'm a bit on delay because it's 4, but you can complete. It's not a problem. You complete the questionnaire, the quiz, during the break. I think it's better you to give time to have a break, and during the break also try to complete the quiz. Then you can, instead of spending here 10 15 minutes filling it, then I think that we can stop here. This is the quiz, okay? And you can do it during the break, but just for you to establish to tell you the homework proposed for this unit. Sorry, this is very simple. It's just make a search, at least 5 traits. Okay, choose a crop species or a vegetable, and revise in literature at least 5 traits with high heritability and 5 traits with low heritability of this crop species, okay, or this vegetable. Okay, this is according to the literature review I have done. These, and these traits are quite medium or low heritability, and these, and medium to high heritability traits on this species. List the traits, okay? 5 with high heritability, 5 with low heritability, and also as an annex to the list, please provide the literature you based your searching, and to list these traits. Okay, it's understand. Anyway, I will copy and paste in the chat as well for you. And now it's time to stop here the unit 3. I'm leaving you the questionnaire here, the quiz. You can fill and send to me and Petra, please. Don't forget Petra for double checking. And we will restart at 4:30.